Now let's talk about viral vaccines. Many argue that vaccines are one of the greatest achievements in modern medicine because of their ability to reduce incidence and prevalence of disease. They play an extremely important role in public health and are responsible for saving millions of healthcare dollars every year. There are three types of vaccines Step 1 wants you to be familiar with: live attenuated, killed or inactivated, and subunit vaccines. Live attenuated vaccines are literally what they sound like. They are live vaccines that are attenuated or weakened through various modifying processes. Some advantages of live attenuated vaccines is that they result in more long-term and durable immune responses. This is because they activate a stronger immune response by leading to humoral and cell-mediated immunity. Cell-mediated plus humoral immunity. These vaccines typically do not require a booster. However, there are disadvantages to these vaccines. The risk with live vaccines is that they're still alive. So there still remains a theoretical risk that these live vaccines can mutate and revert back to their active and virulent self. Now that is scary, but in reality, this is only a theoretical risk, and it doesn't prevent us from using these vaccines in immune-competent hosts. But because of this risk, we do not give live attenuated vaccines to immunocompromised people or individuals in close contact with immunocompromised patients. Can you tell me some populations of patients who are considered immunocompromised? There's three important populations, including patients with AIDS, patients that are on chemotherapy, and patients who had an organ transplant and are on long-term immunosuppression. The live viral vaccines include smallpox, yellow fever, rotavirus, chickenpox, polio, the Sabin vaccine, MMR, and the intranasal influenza vaccine. A mnemonic from the first aid that helps us remember the vaccine is live, one night only. See small, yellow, rotating chickens get vaccinated with Sabin and MMR. It's incredible. I know this is a pretty extravagant mnemonic, but hopefully it will help you remember the different vaccines that are live and attenuated. The killed vaccines contain viruses that are killed by modifying processes but still have some immunogenic antigens present. These antigens can either be viral capsid or envelope proteins that incite an antigenic stimulus in our immune system. Basically, these vaccines have dead viruses that can't replicate, but still have proteins that can be recognized by our immune system. In comparison to live attenuated vaccines, the immune response to killed vaccines is predominantly humoral or antibody mediated. This means that the immunity is not as strong and it's not as long-lived, and therefore it requires booster vaccinations to maintain. Although they don't lead to as robust of an immune response, the benefits of killed vaccines is that they are safer, especially for immunocompromised patients. Killed or inactivated viral vaccines include rabies, the intramuscular influenza vaccine, the polio Salk version of the vaccine, as well as hepatitis A virus vaccine. The last type of vaccine we will talk about is the subunit vaccines. A subunit vaccine is one that is made up of a subunit or a portion of the virus. The subunit is typically a capsid or envelope protein. Essentially, scientists took the virus, broke it up into its basic pieces, and isolated one of those proteins or pieces in the lab. The protein is then mass-produced, put into a vial, and injected into patients. This introduces the patient's immune system to a piece of the virus, leading to a T helper T cell type 2 response. The two subunit vaccines are the hepatitis B vaccine and the human papillomaviral vaccines. Let's talk now about the genomic properties of viruses. Guys and girls, this is where the money is for step 1. The genetic material of a virus is the key way in which we classify them. Remember, it is very plausible for board examiners to present a virus clinically and then ask you to identify it based on its genetic material alone. So let us start with the basics. Can someone tell me from biochemistry, what are the two types of nucleic acids? I hope we are all saying DNA and RNA. We can see from this image that RNA is single-stranded, while DNA is double-stranded. This mostly remains true in virology, except for a few exceptions which we will go over together. So there are two types of viruses, DNA and RNA viruses, and we will start with the DNA viruses. There are 7 DNA viruses, and they are all happy viruses. They include the hepatinovirus family, the herpes, adeno, polyoma, papilloma, parvo, and poxvirus families. They all are double-stranded like the DNA in our cells, except for pyrovirus, which you can remember because it sounds like it is part of a virus. They are all icosahedral, except for poxvirus, and they all replicate in the nucleus of human cells, except for poxvirus again. So now let's talk about where DNA viruses replicate. Remember, the key concept in virology is that viruses rely on other cells to replicate themselves. DNA viruses replicate in the nucleus of our cells. Remember, the genetic material of the virus, whether it is DNA or RNA, is responsible for encoding the proteins required for survival and proliferation of the virus. So with that in mind, thinking back to biochemistry, do you remember in which biological process protein is produced? That would be during mRNA translation. Very good. So, for DNA viruses, what does it take for the DNA to be turned into protein? Well, that brings us back to the famous central dogma of biochemistry and molecular biology. DNA must be transcribed into mRNA, and that would happen where in the cell? In the nucleus, exactly. And once mRNA is made, what happens next? It is translated into protein. Very good. And where does that happen? That happens in the ribosomes found in the cytoplasm. This is the key here. DNA viruses need to make it to the nucleus in order to replicate its genetic material and to produce the protein needed to make it a complete 3-component virus called a virin. But fortunately or unfortunately, medicine biology are all about exceptions. The pox virus is the one DNA virus that does not replicate in the nucleus. The pox virus actually replicates its DNA in the cytoplasm of the cell. Therefore, what do you think the pox virus needs to be able to do this? Well, remember, if the virus doesn't go to the nucleus, it needs to find some way to both replicate its DNA and transcribe its DNA into mRNA, because remember, all those enzymes are found in the nucleus. So what does that mean? Well, it means that the pox virus carries its own DNA-dependent RNA polymerase. It carries its own polymerase that is DNA-dependent and produces RNA. This is essentially the exact polymerase we have in our own nucleus that does mRNA transcription, and it allows this pox virus to bypass our nucleus completely. So now time for a flash quiz. To be infectious, on which viral genetic mechanism does hepatitis D virus rely on? Remember, it relies on complementation. Hepatitis B protein complements hepatitis D virus so that it can survive and be infectious. Now let's talk about the RNA viruses, which are less straightforward than the DNA viruses. In contrast to the DNA viruses, RNA viruses all contain single-stranded RNA, except for the reovirus, which is double-stranded. Once again, here is another exception to the rule. And thinking back, do you remember what is the only single-stranded DNA virus? The only single-stranded DNA virus is parvovirus. Remember, it is part of a virus. And what is the only double-stranded RNA virus? It is reovirus. Very good. So as a learning strategy, it is important to understand the general concepts and then remember the exceptions to the rules. This approach helps us keep in mind the most important concepts while also appreciating that nothing is 100% one way or another. The RNA viruses can be either positive or negative sense, and they can take on one of three forms we mentioned earlier in the lectures. They could be naked or enveloped, icosahedral, or helical. Do you remember back to the DNA viruses, are they mostly icosahedral or helical? Remember, most DNA viruses are icosahedral. And what is the one exception to this? Remember, the pox virus has a complex protein structure that is neither icosahedral or helical. Very good. And another rule to remember is that all helical capsids are enveloped. Now we're going to discuss the difference between positive sense and negative sense RNA viruses. This is a very high-yield concept, and it can be a little confusing, so let's go through it slowly. Like we've said earlier, single-stranded RNA can either be positive sense or negative sense. In order for this RNA to be translated into protein, it needs to be in the proper form. So to make it simple, positive sense RNA molecules are mRNA molecules. Positive sense RNA molecules are mRNA molecules. This means that they are directly translated into protein by ribosomes, just like the messenger RNA is supposed to. So let me say that again. Positive sense RNA viruses are directly translated into protein, just like messenger RNA is supposed to. And where in the cell does this happen? Well, remember, translation is carried out by ribosomes that exist in the cytoplasm or on the rough endoplasmic reticulum. Very good. Here is a list of all the positive-stranded RNA viruses. We have the retrovirus, togavirus, coronavirus, hepavirus, calicivirus, and the picornavirus. A mnemonic in first aid to help us remember them is, I went to a retro toga party where I drank flavored Corona and ate hippie California pickles. Hopefully we can use this to remember that retrovirus, togavirus, flavivirus, coronavirus, hepavirus, calicivirus, as well as picornavirus are positive-stranded RNA viruses. In contrast to positive-sense RNA, negative-sense RNA viruses are complementary to messenger RNA. Therefore, before it can be translated, these RNA molecules need to be converted into positive-stranded RNA, and then they can be properly translated into protein. So let me say that again. Negative-sense RNA must be converted to positive-sense RNA, and then translated into protein. Do you remember what enzyme would convert RNA to RNA? Is that something that eukaryotic cells or human cells can do? Convert RNA to RNA? Well, no, that's why it's so cool. Negative-stranded RNA viruses must carry with them an RNA-dependent RNA polymerase, a polymerase that uses RNA to make more RNA. Only these viruses do that. Now that is so awesome. So in review, negative-sense RNA is converted by RNA-dependent RNA polymerase into positive-sense RNA, which as we said is equivalent to messenger RNA, and then it is translated into its functional proteins in the ribosomes to form a complete virin. So in review, negative-stranded RNA viruses must have or carry with them this RNA-dependent RNA polymerase to make the protein and to complete the virin. In general, RNA viruses replicate in the cytoplasm. Remember, RNA viruses are already RNA and do not need to go to the nucleus like DNA viruses to be transcribed. So the RNA is readily translated to protein, which is then used to complete the virin, and that all occurs in the cytoplasm. But really, Kevin, do you think all of them replicate in the cytoplasm? No way, that would make our lives way too easy. Obviously, there are some exceptions to the rule. Influenza virus and the retrovirus, such as HIV, replicate in the nucleus, but all other RNA viruses replicate in the cytoplasm. Now let's discuss the viral envelopes. Do you remember the two important functions of the viral envelope? Well, one, it aids in host cell invasion of the virus, and two, it protects the virus from our immune system. So another question for you, are envelope viruses more sturdy than naked viruses? So you may think that since envelope viruses have an extra layer, they are more stable. But in fact, this is not true. Envelope viruses are more susceptible to damage and destruction in the presence of detergents, extreme pH, extreme temperature, and organic solvents such as ether. Naked viruses, on the other hand, are more stable in these environments because they do not have a lipid bilayer that is destroyed in these conditions. So in clinical application, when wiping down a surface with sterile wipes, which viruses are more easily removed, naked or enveloped? Well, you got it. It was the envelope viruses. So do you remember where the envelope viruses get their envelopes? Well, as you can see in this image, envelope viruses acquire their envelopes from the plasma membrane of the host cell. When the virus infects the cell, it releases its enzymes and genomic material, and it begins replicating its material. Once it produces its important proteins and is ready to exit the cell, the virus packages itself to its capsid protein, and then, when it's leaving, it takes part of the plasma membrane with it. The exception to this is with the herpes viruses, which actually gets their envelope from the inner nuclear membrane. So now how do we remember which viruses are enveloped and which are not? Well, we can always trust the creative authors of First Aid to provide us with some handy-dandy mnemonics. In total, there are 8 naked viruses, 4 are DNA and 4 are RNA. The DNA viruses include the papilloma, adeno, parvo, and polyomavirus. Think of pap and think of the pap smear. The 4 RNA viruses are calicivirus, picornavirus, reovirus, and hebivirus. Think of CPR-H. So, let's give pap smears and CPR to naked herpes. Now time for a flash quiz. Which 2 RNA viruses replicate in the nucleus? The influenza viruses and the retroviruses are the 2 viruses that replicate in the nucleus. Remember, the retroviruses reversely transcribe their RNA into DNA, and then it's integrated into the genome of the host cell. In order to do this, the virus needs to be in the nucleus and replicating. A good way to learn virology is to remember some general rules that apply to each group of viruses. Once you know these general rules, then you can memorize the exceptions to the rule. There are 4 rules I want you to know about DNA viruses. 1, they are double-stranded. 2, they are linear. 3, they are all icosahedral. And 4, they replicate in the nucleus. Now for the exceptions. Exception to rule number 1 is parvovirus. Parvo is the only single-stranded DNA virus. There are 3 exceptions to rule number 2, and that is papilloma, polyoma, and hepadna viruses. These viruses do not have linear genomes. Papilloma and polyoma are supercoiled circular genomes, while hepadna virus has an incomplete circular genome. The exception to rule 3 and 4 is the pox virus. The pox virus family is not icosahedral, it has a complex structure, and it does not replicate in the nucleus. Rather, it replicates on its own in the cytoplasm.